Hey, so my name is Mimi. I'm a pediatric neuro-oncologist at the Dana-Farber in Boston, and I'm going to give you an update today on our ongoing project looking at MYB-QKI rearrangements in angiocentric glioma. So, as you all heard from David's wonderful talk this morning, the recent explosion of genomics has really revealed a lot of insights into the biology of low grades, in particular the alterations of the MAPK pathway. However, we've all had patients in clinic with the rarer subtypes where we haven't found a BRAF alteration. And the reason for that is that the first few genomic landscape papers were not really powered to find alterations that occurred with a frequency of less than 10%, and weren't really powered to be able to associate those less frequent alterations with histological subtypes. So to address this, we performed a meta-analysis where we downloaded all of the previously published sequencing data, all of which David described this morning, and added our own unpublished sequences to it. And what we ended up with was a cohort of more than 200 tumors, of which more than 150 underwent whole genome sequencing. A number of them also had matching transcriptional profiling. When we then did the significance analysis again with the larger cohort, we were now able to make some new observations. First, we were able to confirm that the BRAF alterations are the most frequent in low grade, and really associate them with the pilocytic astrocytomas. We've color-coded the alterations here, so where you see orange, it's a structural alteration, where you see blue, it's a point mutation, and this is the second message that I want to give to you from this co-mute plot, which is almost 75% of all the alterations we detected in this cohort were structural. So that has implications when you're designing your clinical assays. Assays that rely just on point mutations will not detect most of them. And now we are also able to really try and work out the incidence or the frequency of the less frequent alterations. And actually in our cohort, which we had enriched for grade 2 low grades, MYB alterations are actually the second most frequent alteration. And we're able to associate these alterations with histological subtypes. And in particular, we were interested in this group of tumors here, which we marked as yellow, almost all of which had a recurrent alteration involving a rearrangement involving MYB and QKI. So angiocentric gliomas are an interesting tumor in their own right. They didn't even have a WHO criteria until 2007. They're WHO grade 1 tumors. They're very slow growing. They commonly occur in the cerebral hemispheres, and the kids present with seizures. And the surgical treatment is the main treatment. They form along the blood vessels and frequently associated with pseudo-rosettes that are seen in the ependymoma. And as you know, the treatment for ependymoma is very different to a low-grade glioma, so having a biomarker to differentiate these two tumors is quite interesting. We were also very interested in this rearrangement, not just because of the diagnostic implications, but because it involved two genes that are commonly associated with cancer. The first, MYB, the hematologists in the room may know this gene very well. It's long been associated with leukemias, point mutations, and truncations are oncogenic, and increasingly we're finding alterations in solid tumors. And indeed, when we look at TCGA analysis, we find that MYB is commonly amplified in esophageal and gastric carcinomas. You can see here red on the heat map denoting amplification. As neuro-oncologists, we weren't really I wasn't that aware of the oncogenicity of MYB in the brain. And one of the main reasons for that is that MYB is not expressed in normal postnatal cortical brain where these tumors occur. So I'm showing you RNA-seq data here from Project GTEx, which is autopsy samples, and you can see a very low level of expression of MYB in the cortex where these tumors occur relative to other tissues. In contrast, the fusion partner of this rearrangement, quaking, is actually named because of its essential role in the brain. So QKI includes quaking, which is essential for oligodendrocytic differentiation, and when you knock it out in mice, they can't make myelin, so they literally quake. QKI or quaking has been postulated to be a tumor suppressor, and indeed when we go back to our TCGA analysis, we see that QKI is significantly deleted across adult GBMs. And here you can see a very focal deletion centered on QKI, and it's quite dark blue, suggesting that it's a homozygous deletion. While adult GBMs have a homozygous deletion of QKI, we observed a hemizygous alteration of MYB-QKI in the angiocentrics. So here is whole genome sequencing from 3 tumors, and you can see the MYB and QKI locus, and you can see loss of 1 copy, suggesting that these rearrangements have 1 intact allele and 1 rearranged. All of the rearrangements that we observed had the common pattern that the 5' end of MYB was retained, there was truncation of MYB, and we observed a consistent breakpoint in QKI. QKI is an RNA binding protein, and all of the rearrangements were intronic between exon 4 and 5 and were predicted to be a loss of function for QKI. RNA-seq data of the angiocentrics confirmed expression of an in-frame MYB-QKI fusion protein, retaining the DNA binding motifs of MYB, the transcriptional activator of MYB, loss of the negative regulatory domain of MYB, and also loss of the RNA binding motif of QKI. So we were interested in three main questions. We were interested in wanting to know whether the MYB-QKI fusion was oncogenic in angiocentric glioma. We were quite interested in the observation that MYB is not normally expressed in normal brain, and we were interested in how we might get aberrant expression. And we were also interested in how a tumor suppressor may also be contributing. So to address the first question, we overexpressed MYB-QKI, the fusion protein, 2 different splice variants in mice neural stem cells, and observed in vitro a significant increase in proliferation compared to EGFP controls. And when we injected these cells into mice brains, it was sufficient to induce tumor formation. Now, MYB is a transcription factor, and when we overexpressed it in our neural stem cells, we were able to confirm activation of MYB signaling. And to further investigate its role as a transcription factor, we performed chip sequencing where we pulled down MYB-QKI and also HVK27, the enhancer mark, and observed a strong correlation between MYB binding to enhancers, suggesting it was acting as a transcription factor. We were really interested in this observation that MYB is not normally expressed in normal brain. When we looked at the BRAF altered tumors in the same cohort, we also saw no evidence of MYB expression, so this is the normal. But in all of the tumors in which we observed a rearrangement, there was aberrant expression of MYB-QKI, and very interestingly, this aberrant expression at an exon level corresponded to the breakpoint of the rearrangement, suggesting that it was being driven off the rearranged allele and not the wild type intact allele. To further investigate how this aberrant expression was being achieved by the rearrangement, we first went and looked at the regulatory enhancer landscape of normal brain from ENCODE. MYB, which is not normally expressed, is not normally associated with any enhancers, whereas quaking, which is so essential in the brain, is very enhancer-rich. And we predicted that this MYB-QKI rearrangement would approximately translocate these enhancers to within 15 kilobases of the MYB promoter. And when we performed chip sequencing of actual low-grade gliomas, so on the top track here we're showing a BRAF-driven tumor, and the bottom is an angiocentric with MYB-QKI. Both tumors we observed many enhancers and superenhancers associated with QKI, since it's so important in brain. The BRAF-driven tumor has no enhancer associated with MYB, whereas we observe an aberrant enhancer at the MYB promoter of MYB-QKI tumors and a proximal translocation. And when we performed functional assays, we were able to confirm that these enhancers were able to activate the MYB promoter and that the fusion itself is also able to act on its own promoter. Then finally, we were interested in the disruption that we observed, the hemizygous disruption of QKI. When we looked at exon-specific data of QKI, we observed a partial suppression of expression of quaking relative to BRAF-driven tumors, consistent with a hemizygous loss. And when we reproduced this in our neural stem cells, intentionally using shRNAs to partially suppress QKI, we observed a significant induction of proliferation in cells that also overexpress MYB-QKI compared to EGFP or truncated MYB controls. So if we put all this together, we have an example here of one single driver rearrangement. Now, I think for pediatric oncologists, this is quite interesting because we frequently see tumors that are characterized by one single driver rearrangement. This rearrangement results in enhancer translocation, causing aberrant expression of MYB in a context where it's not normally expressed. This MYB-QKI fusion then further activates in a forward feedback loop, driving its own expression, and we also get simultaneous loss of a tumor suppressor. So in summary, we have found that MYB-QKI rearrangements define angiocentric glioma. That I think has quite a lot of diagnostic and therapeutic implications. I think that this finding is probably going to be relevant to many other pediatric solid tumors, and as I said, there's diagnostic and therapeutic implications. And then finally, I am one of the very lucky people that gets to go to work every day and work with people that absolutely inspire me. I really want to acknowledge my mentors. I'm in the Means Lab, co-mentored by Keith Ligon, and I was so lucky to move to work with Mark 5 years ago. And as you can see, this is a collaborative project with lots of people, and I'd like to acknowledge everyone, and also the team from Chuck who worked on this with us.